Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Good morning everyone. Welcome back to my channel Pari's tutorial. Last class we have already gone through centrifugation and all. And in this class I want to discuss about the technique called chromatography. What is chromatography? Chromatography, chroma means color, right? So it's a lab technique that is mainly used for the qualitative and quantitative analysis of the mixture. So when you get a mixture of sample, so based on the type of solvent and based on the type of stationary phase, how the affinity towards them is regarded as a principle for this chromatography. Okay, so used for the separation of mixed compounds into individual components. So okay, when you take a sample, you will once up after the separation, you will the affinity, like how they are stationary phase and how the particles are traveling with the mobile phase, how the affinity is there between those two phases is what you call is chromatography. Okay, so main principle here is different components in a mixture travel with different velocities and affinities on the stationary phase. Okay, the components of mixture get retained at different lengths of time, right? So based on how far, how long the affinity is there between, how the velocity of that particular molecule with that stationary phase and mobile phase decides your retention time. Okay, this is what is called a chromatography. Okay, what are the types of chromatography? So you should probably heard about the paper chromatography, which is a basic simple chromatography. So okay, and the next one is a thin layer chromatography, column and gas. So column is again divided into many things like affinity chromatography, ion exchange chromatography, gel permeation chromatography and all. Okay, I will discuss the main basic principle about it. And chromatography contains a paper mainly of Wattmann's #1 filter paper is generally used. Okay, and that particular paper is made up of alpha cellulose and a beta cellulose. Okay, so stationary phase in the paper chromatography is the pores in the stationary phase act as the pores, or the water present inside the pores of that particular paper acts as a stationary phase. Okay, and the solvent that we are using acts as a mobile phase. Okay, so it's just, if you just place an ink on the paper, it would immediately spread, right, based on its affinity towards your stationary phase. It would interact with the water molecules present in that particular paper. In the same way, the paper chromatography also relies on same thing. So made for the separation of amino acids, chlorophyll and DNA bases. Paper used is, I already told you, it's Waterman's #1 filter paper. Okay, it's mainly used for the paper chromatography. So it is composed of alpha cellulose and beta cellulose. I told you the water molecules present in the pores would act as a stationary phase. So right, so when you just place an ink drop, or when you just place one small colored substance on a paper like Waterman's filter paper, it would gradually, and placed it in the solvent or something like that, or in the water. For example, I'm just taking it as a home example. Okay, when you just place an ink drop and you just add water to that particular ink drop, so based on the absorption and based on the affinity and the reaction, the particular ink drop would travel to further length, and you'll get different colors on it, right? Bands or different colors line. So that is how you see paper chromatography. Okay, the sample to be separated is added with a capillary tube or a syringe tube. So this is what important is. What is RF? So RF is the distance the spot has traveled and the distance has the solvent has traveled. So when you place this particular paper in a solvent containing the mobile phase, it could be ethanol, it could be methanol, it could be ether, it could be chromoform, like that. Okay, and when you place a sample containing all the components on the filter paper and place it on the mobile phase, slowly the mobile phase would start moving. The paper would start absorbing that particular mobile phase, and based on it, it will give you the distance that has been traveled. So you will get a small horizontal line where the solvent has traveled. In the same way, you will get a small band like structures where the spot has traveled. So based on it, you can decide how much the substance has traveled and how much spot has traveled. This is the, based on it we can decide the RF value. So if you take a compound which is a pure compound, like it's a polar compound which is a pure and stronger compound, it takes time for you to get spreaded, right? So it's relatively having a low RF, right? If you take a non-polar compound which contains water in it, or it is a diluted sample, and generally the diluted sample move faster, it has got affinity with water molecules in the stationary phase and it moves faster. Okay, so the RF would be at a higher than the polar compounds. So that is the main basic principle involved in paper chromatography. Okay, so I told the paper chromatography, I told you that is how, see, this pot has moved only for 2 centimeters, this purple has moved for 8, and the solvent has moved to 10 centimeters. Based on it, we can decide the RF value. So RF value will generally be always lesser than 1, right? Even for the TLC, even for the thin layer chromatography also, the RF would be generally less than 1. Okay, so this is the paper chromatography. What is thin layer chromatography? It's almost the same like paper chromatography, but it has got an absorbent layer attached to the glass or a plastic side. So what happens is, in thin layer you will be applying the adsorbent with on a thin layer glass plate. That's why it's also called as adsorption chromatography. Okay, here a glass plate or glass slide or plastic surface is taken, and onto which you will just add the absorbent material. The absorbent material would be like alumina or silica, which is maintained at 0.1 mm thickness. Okay, you'll just apply it and you will just you should be making it sure that all the water molecules, all the residue should be left over, right? It should be kept in the oven for some time, so that once it is completely dried out, you can go for the further process of placing it in the mobile phase. Okay, so I told you RF value generally are less than 1. So this is thin layer chromatography. So when you play how far the molecules have traveled and how far the spot has traveled. So based on this, we can just derive the answer. Okay, this is generally less than 1, I told you. Okay, so this is thin layer chromatography. So what is column chromatography? This is main important thing and most interesting thing in chromatography. See, what is column chromatography? In this column chromatography, you will be having a column which is around 25 to 15 to 25 centimeters. Particular column is filled with the stationary phase. It could be anything like alumina, silica, or something like that, which is called a matrix or something. Okay, and that particular thing acts as a stationary phase, and to which you will just add a sample, and over the top you will be pouring the mobile solvent. Okay, so what is column chromatography? Column chromatography is the particular solvent comes along with the mobile phase. So mobile, when you add the mobile phase, based on the gravitational pull, the mobile phase would take the sample down towards the stationary phase. So the based on the interaction of that particular sample with the stationary phase in the column and the mobile phase coming from the top would decide the sample's retention time. Okay, the sample's retention time would be depending on that. So for example, so I will tell you. See, this is, I will tell you high pressure later. Okay. Okay. So in what I'm here to talk about is, okay, I will tell you better with the uh, diagram or something like that. See, for example, if we talk about first, what is column chromatography? Column contains, this is the length of the column. It would be around 15 centimeters, 10 to 15 centimeters. Okay, it has got a diameter also. Okay, and generally the stationary phase will be coated here with the alumina or silica, or at 0.1 to 0.3 millimeters. So that is a thickness that we generally use for the stationary phase. Okay, so it has got a pump here so that you can get all the illusions collected. Okay, so you will take the sample at here. Okay, and the mobile phase is to be added. I told you mobile phase may be ethanol, ether, methanol can be added. Okay, so the sample now reacts with the mobile phase that is added, because the mobile phase moves in the gravitational force towards down. Okay, so when the sample along with the mobile phase reaches to the stationary phase, based on the interaction with the sample with the mobile phase and the stationary phase, you will get your substance collected. For example, I told you, based on the type of stationary phase, you will get either silica or aluminum adsorption chromatography. For ion exchange chromatography, ligand, gel beads. See, what are all these things? So, ion exchange chromatography is mainly dependent on the oppositely charged particles. For example, if the column contains the negatively charged, okay, then you have to add the positively charged components, so that it would definitely would be higher, and you will get the substance to be separated at the last. You will be adding some inhibitors so that they can be removed and you will get your substance out. So that is what you call ion exchange chromatography, where the oppositely charged molecules would happen. See, the stationary phase should be having a phase that is opposite to the charge of your sample. So if the sample is positively charged, coat the column with the negatively charged. Like, you should be adding the resin, like ion exchangers. I'll tell you what are ion exchangers also. Okay, so they should be added with that. And the stationary phase is also known as ion exchanger. I told you, used for separation of proteins, peptides, nucleic acids and all. So what is cation exchanger and what is anion exchanger? Okay, so you all know that cations, they are positively charged particles. They move towards the cathode. Generally in electrophorese we have already gone through. So cation exchanger is negatively charged, is made negatively charged, so that the positively charged cations in the sample can be attracted. In the same way, the anions are generally negatively charged and they move towards the anode in electrophoresis. So these anions are known positively charged and attract those anions negatively charged sample towards it. Okay, that is what you call the anion exchanger. So what are the anion exchanges and cation exchanges? I told you the carboxyl, carboxymethyl and sulfomethyl are generally used as a cation exchange. I told you they are negatively charged, so they attract only the positively charged particles in the sample. In the same way, the anion exchanger would generally have a positive charge, so that it would attract only the oppositely charged and negative particles for the separation in the sample. Okay, they are the buffers used. So they may ask you what are the buffers used. So acetate, phosphate, these are all the buffers. Okay, I told you the buffers are all acetate, phosphate, lactate. Okay, even in the anion exchanger, like tris and the pyridine are generally used as buffers for the anion exchanger. Okay, so this is the cation and anion exchanger. These are one type of column chromatography. Okay, what are the other types of column chromatography? The other type of common chromatography comes with ligand chromatography, sorry, affinity chromatography and gel permeation. Okay, so let's talk about affinity chromatography. So what is affinity chromatography? Take, like, if the sample contains enzyme, so you have to separate that particular enzyme. So what you have to do, the column should be coated with a suitable substrate. So you all know that enzyme substrate is a lock and key type of model. Like, if an enzyme is like this, the enzyme substrate should be like this. Okay, it's a lock and key type of model. You know that enzyme substrate reactions are like that only, because it has got a maximum affinity towards it. So if you have a sample containing that particular enzyme, please, you have to just code that particular column with the substrate suitable for that enzyme. So what happens when, for example, feel this is the column that I'm talking about. Okay, and you have just coated with all types of substrates like this. So you got samples, but what particular sample would bind towards your substrate? The remaining get separated at the back. So finally, at the last, increasing your pH or increasing your ionic strength last. So this is what your receptor is one important thing, like enzyme substrate only. Okay, so if it is like even the nucleic acids and proteins are also got a nice affinity towards it. So if you need to separate nucleic acids, you have to just coat the stationary phase with a particular protein, because nucleic acid has got a higher affinity towards your protein. Okay, so this is how the affinity is. Though, so okay, let's go for what is HPLC. So HPLC is it's a high pressure chromatography, or it's also called as high performance liquid chromatography. Okay, so it's a modified. I told you this is a modified chromatography which depends upon the pressure that we add from the top. Apart from the sample, apart from the mobile phase, you should also be adding high amount of pressure like 50 megapascal. That particular stainless steel made column should be able to withdraw, or should be able to hold that particular pressure. It should be like a minimum of 50 megapascals. That particular column should be able to hold that pressure. So the pressure that is applied here is a 50 megapascal, and the diameter inside the column is 4.5 to 5 millimeters, and where the peaks are obtained. Yes, I'll tell you what is this. So here discuss. Okay, so what they do is, generally they will inject the sample here, and they will have a high pressure pump here with certain megapascals of pressure to be allowed for the sample for the separation, and based on it you will get the system, and you will get a graph here in the recorder. Okay, so about separation through so let's talk about HPLC separation. Okay, in HPLC separation, let's talk about glucose. Okay. First thing is, in the last electrophoresis classes also I have already told you the samples must be compared with the standard curves. On the electrophoresis you have compared it. In the same way in HPLC also you have to compare the retention time of with the standard, and also the concentration of that particular sample with the standard. Okay, here in HPLC you can first of all know the retention time, and also you can also detect the concentration of that particular sample, of that particular compound in the sample also. Okay, so for example, before you add the sample, so you should also be knowing the standards, right? So for example, so I have added glucose to your column. Okay, and you have run it, and through the detector you got a peak. So peak is around like this. So this indicates a time. So after injecting the sample into the column, you got a peak at 5 centimeters, which says that the retention time for glucose is 5 minutes. So retention time for the glucose is 5 minutes. Okay, in the same way, now go for a new experiment where you have added sucrose. So sucrose, when you add sucrose and you got a retention time, and you will be detecting it in the system, and you got a peak, and there you got a peak at 8 minutes. So this says this is the retention time of sucrose. So sucrose's retention time is 8 minutes, and the glucose retention time is 5 minutes. So these are the standards. Okay, this can be compared with the sample. For example, you got a sample where you have an unknown sample, right? So for example, this is the column, just the unknown sample. Okay, and you have added the mobile phase and everything, and you got a retention time where you will be detected in the system. So for example, so these are the peaks. Example I'm telling you. Okay, see, this peak is not known because it got it immediately after injecting the sample, and you got a peak at 8. I told you the peak at 8 indicates sucrose. You didn't get any peak at 5 minutes, so there is no glucose in the sample. So this is how you detect the unknown sample. You have to compare with the standards and then run it. Okay, this is what is a principle involved in HPLC. Even not only the retention time, you can also detect the concentration also. For example, millimolar concentration can also be detected. So based on the gap between this, the area between the peak and the time. So this is what you call the concentration. At 5 minutes you got a 20 millimolar concentration of glucose. This is a standard. Okay, so if you at that time if you got a bigger peak, like, so this is the same 5 minutes but you got a bigger peak, sorry, much more bigger peak, so obviously the area is more, and this particular area tells the concentration of that particular glucose at 5 minutes. So the concentration of that particular sample, unknown sample, can be detected with the compared with the standards. So here this is the standard glucose retention time for, and also concentration, and when you compare it with the unknown sample, you will be detecting at 5 minutes you got glucose but at a higher concentration, so the sample contains more of glucose. So this is how you analyze your compound unknown sample comparing with the known sample. So this is what is gas chromatography. So before I talk about gas chromatography, you should be aware of what is volatility, what are volatile compounds. So compounds which has got a lower boiling point, like some compounds when they are opened, they immediately get vaporized. So they have high volatility, right? So those volatility compounds would generally have low boiling point, right? So they are inversely proportional. So the main important principle involved in gas chromatography is, so when you are made to make the samples mixed with the volatile compounds, so based on it the separation would happen. Okay, so this is the whole equipment of gas chromatography, and you got an oven here where the column is maintained. This is a stationary phase. Okay, the generally the stationary phase in gas chromatography are mainly coated with silica or with a wax, so that it really tolerates a high amount of temperature, because around 300 degrees temperature is maintained in the oven. Okay, and you get the sample to be injected here. Okay, and the sample to be injected is first of all mixed with the volatile compounds. Okay, those volatile compounds like methanol, you also be knowing about acetone and all, these are having high volatility. Okay, those compounds are to be injected near the port at high temperature around 55 degrees to 60 degrees, because at that higher temperature whatever the samples that are placed in the liquid part would immediately get vaporized. Okay, and it is ready for the separation. Okay, and here the column is the stationary phase itself, and you got a carrier gas like helium or nitrogen are generally used as inert gases for your mobile phase in gas chromatography. So when the sample is injected from the top, and they are made into volatile compounds, at the same time when the carrier gases like mobile phase is added here, it depends upon how the particular compound would react with the stationary phase or with the mobile gas phase. Okay, for example, if you have a compound which has higher volatility, so that would immediately move away from this stationary phase and get detected first, because it has got high volatility, and it interacts more with the mobile phase than the stationary phase. So if the sample contains compound that doesn't interact with the mobile phase and only interacts with the stationary phase, which says that it has got a lower volatility, right? It has got the affinity only with the column, with the stationary phase. So it could be having a higher boiling point, so doesn't react with the mobile phase, it would only react with the stationary phase and get separated at the last. So the main principle involved here is, whatever the what are the components in the sample are having affinity with the column, with the stationary phase and the mobile phase, would be detected in gas chromatography. What happens in gas chromatography? How you detect the peaks in gas chromatography? Same as HPLC also, you will be getting peaks, and based on the current that is generated the last, you'll be getting the concentration and also the retention time in gas chromatography. Okay, so this is the simple procedure of gas chromatography. Hope you like the class, hope you are clear with the doubts and all basic principles are covered in this. So most okay, so thank you and have a nice day. See you in the next class.